Hello everyone. Today we're going to talk about the structure and functional molecules in our body. Okay, start with DNA structure. The first thing that is very important in the structure of the DNA is that it's composed by two strands, twist, forming a double helix, and these two strands are made up structures called nucleotides. These nucleotides are the bricks of our molecule of DNA. We're going to go deep to this structure. A nucleotide. A nucleotide is a molecule made up of a phosphate group, a 5-carbon sugar, in this case called deoxyribose, and a nitrogenous base. There are 4 different nitrogenous bases: adenine, guanine, cytosine, and thymine, that form the DNA. This structure of nucleotide is very important because it's the base of the structure of DNA. So, we have two different molecules, one called nucleotide and the other molecule is called nucleoside. The difference between a nucleotide and a nucleoside is that the nucleoside lack of the phosphate group. Yes, in this case, as you can see, we don't have the phosphate molecule joined to the deoxyribose. Yes, we have instead in the nucleotide the phosphate group. This picture is very similar but with different colors. As you can see, with blue, clear blue, we have the nitrogen bases called adenine, and we have our sugar called deoxyribose. Yes, but these two components for a nucleoside. If we put or we join or we bond a phosphate group to this molecule, we're going to have a nucleotide. This is a nucleotide of adenine because adenine is the only component that changes because we have 4 different bases that form the DNA. So, the basic structure of the nucleotide, because we have 2 different nucleic acids, we have the RNA and the DNA. There are a simple difference between the structure in the nucleotide of the RNA and the DNA. This structure, or this difference in the structure, is that the sugar, the ribose in the DNA, lack of oxygen in this bond. As you can see, the RNA has the oxygen and the hydrogen. In this case, we don't have the oxygen. This is because this is called ribose and this is called deoxyribose. Very important because to make molecule acids or nucleic acids, we need a sugar. In this case, we are talking about the DNA. So we need the deoxyribose joined to a phosphate group that's going to be the same everything, and we have a nitrogenous base. The bases that we found in the DNA are cytosine, we have thymine, we have guanine, and we have adenine. The uracil, as you can see in this picture, belongs to the RNA molecule. The RNA, instead to have thymine, it has RNA. So let's clear everything. Important, as you can see in this picture, the guanine and purines are the guanine and adenine are called purines. Purines are larger than pyrimidines. It has 2 rings, one ring a second ring, while the pyrimidines has only one ring. So pyrimidines are smaller or purines are larger. It depends of the of how do you see this in context. Okay, let's continue. We have again the structure, but in this case it's simpler, about the purines and pyrimidines. So it's important to understand that purines have a double ring structure, yes, because have 1 and 2 rings, in this case 2, 1 and 2 rings, while the pyrimidines only have 1 ring, a single ring structure. As you can see, we can see only 1 ring in each structure or molecule structure in this basis. This is very important to understand how the bases pair between them. So let's going to clear everything. And continue. We have the hydrogen bonds. There are bonds very special because these bonds keep the 2 strands of the DNA together. Hydrogen bonds are these bonds that are established between the nitrogenous bases. So, because each nitrogen bases are bound with a hydrogen, they're going to share an atom of hydrogen between them. So in this case, the purine, as you can see, I'm going to share with the color. As you can see, this hydrogen belongs to the purine, yes, and the hydrogen in this part belongs to pyrimidine. What happened? The pyrimidine going to share this hydrogen with the purine, as the same thing that the purine going to share these 2 hydrogens with the pyrimidine. So they're going to hold together these hydrogen atoms. This is because we say that it's a hydrogen bonds. In Spanish, we could found that is called puentes de hidrógeno. So, with respect with this other picture here, we're going to realize that cytosine and guanine establish 3 hydrogen bonds, while thymine and adenine establish only 2 hydrogen bonds. This is very important, and this is the reason because the bond between cytosine and guanine is more established than the thymine and adenine. In the DNA structure, we're going to find that always the cytosine join with the cytosine join with guanine, and the thymine always join with adenine, always, because if occur a mistake, maybe could have this process, but this mistake is going to be called mutation. We're going to talk about this about mutation later, but it's important to understand that always we're going to pair cytosine and guanine and thymine and adenine, always. Okay, we have the DNA strand orientation. This is another thing very important. Why? Because we have that one strand goes to the 5' end to the 3' end, and the other strand starts with 3' end and finally with 5' end. What that means is very simple. The carbons in the sugar has a number. I'm going to clear these structures, has a number. I'm going to write with red. This is the first carbon. This is the second carbon. Sorry for the numbers. This is the third one. This is the fourth. And as you can see, this is the 5. Yes? So, what happened? Always, when we are talking about the DNA, the third carbon joined to the phosphate of the previous nucleotide. Yes? And the phosphate of the current nucleotide joined with the 5 carbon. This is because we call 5' end and 3' end. What that means? That in this case, yes, we don't have another nucleotide in this part. Yes, so we're going to say that this bond is free because only have the phosphate group. But if you can see at the end, the third carbon in this part, yes, is free. It don't have the other nucleotide in this part of the structure. This is very important to understand the orientation. This is because this line runs in this way and the other round in the opposite way. So always the beginning of the strand is going to be the 5'. Always the beginning of the nucleotide is going to be the 5' end. So that means that the beginning is where it starts with a phosphate group joined to the 5th carbon in the sugar. While in this case, we're going to say that it starts with the same point, yes, in the 5' because the phosphate is joined to the 5th carbon in the sugar. But in this case, we're going to realize that the head, in this case, for example, the head of the strand is opposite to the other strand. This is because if we turn around this change, we're going to realize that we're going to have the phosphate groups here and we couldn't join or bond the bases between them. So this is important, the orientation, because we need to have face to face the strand in order to bond, join, link together the different bases. Yes, as you can see, adenine and thymine, thymine and adenine, cytosine and guanine and cytosine. This is very important and we call this like a anti-parallel orientation. Anti-parallel means that one strand runs in one way and the other runs in the opposite way. Yes? So let's continue. The two strands in the double helix are complementary too. What means this complementary word? That the cytosine and the guanine always going to link together, complement between each other, and the thymine and the adenine is going to join together always thymine, adenine, thymine, and always cytosine, guanine. Yes, this is important. Why? Because the way that we establish the hydrogen bonds is very important. So in this case, it's very difficult to join the adenine with the cytosine. It's not easy because the structure is not the same. So this is because when the adenine joins with a cytosine, we call a mutation, that is a mistake that occurs in the replication of the DNA. We're going to start with the function and molecule. I already talked a little bit about the RNA. RNA is the ribonucleic acid. This molecule is present in all living beings, as a material generic in retroviruses or perform another function in the other organisms. Retroviruses are viruses that the genetic material is the RNA. Example of retrovirus, we have the human papillomavirus, we have the human immunodeficiency virus, and we have the coronavirus. The function of the RNA is act as an intermediary between the DNA and the proteins. So, in this case, we're going to talk about a type of RNA called messenger that carry the message that is codified in the DNA to be produced proteins in the ribosomes. Sometimes this RNA can be catalyst. Catalyst is a substance that increases the speed of chemical reaction by reducing its activation energy. So they are very important in chemical reaction. As you can see in the picture, you're going to realize that this is not a double strand, they only have one strand. So, RNA versus DNA. It's a basic unit of ribonucleotides. So, ribonucleotides are the basic units in both molecules. The difference is we have uracil in their RNA and we have thymine in the DNA. In the RNA, this uracil is going to join with adenine, and in the DNA, the thymine is going to join with the adenine. So in this case, depending on the molecule, we're going to have a bond between adenine and uracil in the RNA, and we're going to have a bond between adenine and thymine in the DNA. So, continue. They both have the nucleotides, but remember that the difference between the nucleotide of RNA and DNA is the sugar. The RNA has a ribose and the DNA has a deoxyribose. The RNA is a single strand, meaning that only has one strand, different than the DNA that has two strands. And the RNA can be found in the nucleus and in the cytoplasm, while the DNA can be found only in the nucleus. Ribonucleotide versus deoxyribonucleotide. We already talked about this. The ribose has this oxygen and the deoxyribose lack of this oxygen in the molecular structure. This is because they call different, and this is because these molecules perform different activities. Types of RNA. We have a lot of types of RNA, maybe 10, but we're going to focus only in 3 types of RNA: messenger RNA, ribosomal RNA, and transfer RNA. What we're going to focus in this type of RNA is because they are involved in the synthesis of protein. Yes, so they are involved in the transmission of the DNA information in different stages until we produce proteins. Messenger RNA. The function of the messenger RNA is to encode nuclear DNA information and transmit this information to the ribosomal RNA. The ribosomal RNA is the RNA that is inside of the ribosomes. Ribosomes are made up RNA, ribosomal RNA, and have 2 units, the major unit and the minor unit. It's part, the function of the RNA is to catalyze. Remember that A catalyst is a substance that increases the speed of a reaction. So, in the RNA, in the ribosomal RNA, the function is to catalyze the formation of the peptide bonds. Yes, these peptide bonds are very important to to synthesize proteins. And finally, the transfer RNA has a very important function in this process, is because the transfer RNA carry the amino acids needed to produce the peptide bonds in the ribosome. We're going to focus by the moment in the structure and function of the RNA, and in another class we're going to talk about the replication, transcription and traduction of the DNA in order to understand what is the specific function of each type of RNA. So another functional molecule is the proteins, or are the proteins. Proteins are very important molecules in our body because they perform different functions. The proteins are polymers. Polymers is a large molecule or macromolecule composed of many repeated subunits. So, if we're going to return to our example of the bricks, the proteins has bricks called amino acids. Each part of this protein is an amino acid. We could have different amino acids, but they all repeat in the molecule. This polymer is formed by the binding of various amino acids whose sequence is determined by the information encoded in the DNA. So in this case, the sequence that is in the DNA is going to be the same in the proteins. What is important, because if we change this sequence, we could have different proteins. And this is because we say that all the information for the life is in the DNA. Because the DNA has the genes, very little sequences that form different proteins. The different proteins produce or perform different functions in our bodies. The function of the proteins. They are a structural function, yes, as it can be a structural part of the tissues like muscles, tendons, and skin, nails, and other organs. They participate in the muscle contraction. This is important because actin and myosin are the proteins that allow the muscle to contract. They are involved in the transport of oxygen to all organs, while the hemoglobin protein, they degrade substances like catalysts. They accelerate metabolism process as a many catalyst. They could have function of receptors, yes, a structural, a storage of oxygen, yes, or in this case of another molecule. It could be enzyme. This means that the enzymes are catalysts. They could transport oxygen and other molecules. They are very important in the protection of our body by the immunoglobulin. Yes, they carry message like the insulin, and I already said that contract are the main structure that control the muscles. The protein structure. Protein structure is made of amino acids. These balls in this chain are amino acids. Each part of this chain is an amino acid. Yes, an amino acid is composed for this molecule. It could change depending on the amino acid, but all of them stay with this structure. The only thing that have a change in the molecule is this part. It could be different chain of carbons until form different amino acids, but they're going to stay with a group amino and a group acid. Yes, carboxylic acid is called this one. So, what happens? The ribosome catalyzes the process of join to bond each amino acid. The amino acid is carried by the transfer RNA. And when they finish this process, we're going to say that we have a primary protein structure because it's only a sequence of amino acid, it's a chain. Yes? This chain has to be modified in order to perform the function. So, the secondary protein structure occurs when the sequence of the amino acids are linked by hydrogen bonds. So, in this case, they're going to start to twist the molecule using the hydrogen bonds between the amino acids. We have the tertiary protein structures that occurs when certain attractions are present between alpha helix and platter sheets. We have alpha helix hits and we have platter sheet hits, and occurs different attractions, so they're going to twist more the protein. The quaternary protein structure is a protein consisting of more than one amino acid chain. Yes, so in this case, different chains of amino acid is going to join in order to produce a quaternary protein, and this protein is finally the protein that performs different activities or functions in our bodies. Okay, this is all for today. See you in the next time. Goodbye.